have your attention, please? For our talk today, we are very happy to welcome Dr. Michael Martin from Lawrence Berkeley National Laboratories. He's been there for the last 17 years, but comes to us through a chain of educational institutions. He is an undergraduate of San Diego, so here in California, followed by his PhD from Stony Brook University, time spent then at Brookhaven National Laboratory, and obviously then he began a postdoc at UC Berkeley, which he then just transitioned up the road into Berkeley National Labs. We're very happy to have him here today. We're going to be taking our speaker out for dinner after the presentation. If anyone would like to join us, please come and see him at the end. And he's here today to talk to us about spectroscopic imaging with a 100 million watt light bulb, and what we can do with this, which I hope is quite a bit for $100 million. So for that, I will turn it over to our speaker, and if we could welcome him warmly, please. Very much
Thank you very much for the nice introduction. Yeah, it's always kind of fun to come up with titles like this to see if we can catch people's eyes. You know, $100 million is a fair bit to spend on a facility, and so hopefully I'll show you some of the types of things that we can do with this facility. But I'm really going to only touch on one aspect of it. There's quite a lot of other X-ray science and other things going on. My specialty is in the infrared, so I'll be focusing on the infrared part and how we use infrared to be able to do new types of imaging. So I'll start my talk with just a brief introduction as to what is this facility. It's called the Synchrotron Light Source, and so I'll jump into what that is. But just first, this is a picture of it. This is actually a picture up on the Berkeley Hills. If you are anywhere in the San Francisco Bay, oftentimes you can spot this dome structure up in the hill because it's been there for a long time. Lawrence, the person that we're named after, actually built it for one of the Synchrotons that he built back in the 40s, is when this originally turned on. And then the building was repurposed into this Synchrotron Light Source facility in the early 90s. And so we've been running for the past 20 little bit years as a Synchrotron light source, so I'll get to that part. Also, this is just my favorite current picture because a couple years ago when the space shuttle actually was being flown down to Los Angeles to a museum, it did this nice slow tour around all kinds of different places, and the Lawrence Hall of Science is just up the hill from us, and so that was one of the places they went right by, and so they flew right over, and it was really impressive to see this thing go super slow as it was flying by right over. So that's a real picture, not photoshopped or anything, which is always kind of fun. So let me jump in with what is a synchrotron light source. A synchrotron light source is an accelerator that's made to make light. Okay, and so we accelerate charged particles, and so this is like a cartoon inside the building of what it would look like. We accelerate charged particles, and in our case, we just use electrons. So we start with what we call an electron gun. It's basically just a big cathode. It's what used to be in the back of television sets before they were flat screen. You would shoot out electrons from this cathode with just a large voltage to strip them off of a piece of metal, and then you start accelerating them down a linear accelerator. So that's called a linac. And then we go into a booster ring. So this booster ring, its sole purpose is to just get the energy of the electrons up much, much higher. And so we start with electrons that are basically at rest here. We boost them up to what we call 50 million electron volts worth of energy through this little linac portion. And then the booster ring, they go faster and faster and faster. And as they're going faster, we ramp up the magnetic field so that we stay in the same orbit the whole time. And so then we ramp the magnetic field up synchronously as we're adding more energy to the electrons, and we get them all the way up to almost 2 billion electron volts of energy. So now they're going very, very close to the speed of light. They then are injected into the main storage ring. So this kind of pinkish looking color here is the main storage ring. And so as the name implies, we try to store them. We're trying to just keep these electrons going around and around the circle, because as they go around the circle, they're emitting light. So the electrons are whizzing around here. As I said, they're going very close to the speed of light. It's 0.999, I think it's 7 nines the speed of light. And so when you get to that kind of speeds, relativity kicks in, and so the electrons become very, very heavy. As they're accelerating, they actually start emitting light in a different way than we're used to in terms of what electrons normally emit light. So a simple example is in a radio antenna, for example. you have just a piece of wire, and you move electrons up and down this big piece of wire. That'll make your electric field follow that electrons up and down, and so then that electric field will propagate out in the form of radio waves, is what we mostly know that as. And so radio waves will come out, and they'll go out in this dipole field around this piece of wire because we're forcing the charges to go up and down in that aspect. In this case, because they're highly relativistic, they actually come out in a little bit different pattern. And so if I show it here, what we're using is the property of that whenever charged particles are accelerated, and so in this case the acceleration is just going around a corner is the acceleration that we're using, then they will radiate lights that's very much like this dipole radiation. So dipole radiation usually looks kind of more like a figure 8 if you drew it in 3 dimensions, if you can kind of imagine that. But because of relativity and the fact that we're in a different frame than the electrons put out something that looks like a figure 8, but because they're now really highly compressed, the light actually gets bent all the way down to where these 2 lobes of the 8 go now straight forward. And so this becomes a really nice tight beam of light that comes out. And so this little cartoon here is showing how the electric field and the electromagnetic waves actually come out from these electrons as they're going very close to the speed of light. So this is a very intense and really a good source of light, because you like light that is now well focused coming from a really small spot. That's something you can then use to refocus onto a sample really nicely. So like a laser, like this laser pointer, is a really good coherent source of light, and you can get a nice tight beam that you can send to a long distance and still be able to see it on the other side instead of a light bulb that's just going out in all directions. And so this has some properties that are like that. It's a much tighter beam. We can be able to do a lot more by refocusing the light down on the sample end. Once we get down further down, then that will be able to. Oh, good grief. Let's not show this. So we can then focus that light down. And so the question is why would we go to all this trouble to be able to do this? And so let's talk about brightness for a minute. So brightness is how much light you can focus onto a spot. So how focusable and how many photons you have. Both of those go into this equation. And so if you start with, this is now really looking at the X-ray part of the light. But if you start with things that are down in more normal life sense, you'll have light bulbs and candles and stuff like that that have a brightness that's way down here. These are now orders of magnitude. So every one of these is a factor of 10 higher and higher and higher. So you can go up a fair bit to get to something like the sun. The sun is something that's pretty bright. It obviously lights us pretty well. But its brightness is it's pretty good, right? We can still see that it's a large source. It's not a point source, for example. If we go up to one of these bending magnets, as I showed you here, we're jumping up, 5 more orders of magnitude in brightness. And then if we go to a specialized source where we do not just one bend, but a bunch of bends all together to be able to add these up coherently, we go even further up. And so if you think of how much light you're getting out up here, it's something like 10 to the 9 times what the sun puts out, right? So a billion times higher than what you get from the sun. And again, you can say or 10 to the 9 above an X-ray tube, or 10 to the 13th above a light bulb. So you're getting to just totally new types of sources of light, and so you can really start to drive the science you can do with it into realms that you just couldn't think of doing with any of these kind of more conventional types of sources. And so that's really the gist of what we're doing, is to be able to say, if we have a new type of light source, something that's so much brighter, so many more photons that we can put on a sample, we can really now drive to things that we couldn't even think of doing before. And so that's the main thing that you're getting out when you build a facility like this, is that you get this huge brightness advantage. So the brightness of this light then comes in all different colors of the light, right? And so the bend magnet spectrum, the simplest one where you just have one bend, it actually spans a range that's really quite broad. And so this is the visible piece of the electromagnetic spectrum. It spans the infrared, even gets down into the far infrared or terahertz or microwave radiation on the low end. And then it goes up on the high end, through the ultraviolet, the soft X-rays, even hard X-rays. We don't get up to gamma rays. That's a little bit higher than we're getting to. But it covers this huge range of spectrum that people can then use for different types of science. So the cartoons I showed you, and even this cartoon here, you can see that there's, you put ports of light, at all different places around your more or less circular accelerator. And each one of those can use a different part of the light to do different types of science. And so some of them will be tuned towards the hard X-ray to do crystallography. Some of them will be tuned towards the soft X-ray to be able to do where you're kicking electrons out of atoms, for example, trying to figure out what atoms are there, what chemical bonding is happening. And some of them, which is what I'll tell you for basically the rest of the talk, will use the infrared part of the radiation to be able to now look at vibrations of atoms and look at really different types of things. So all of these have these big advantages that you can really use for being able to do science that you really couldn't do before. And then let me just say, feel free to ask me any questions as we go along, because I'm going to go through this introduction and then I'm going to jump into a whole bunch of examples. So if you'd like to ask me anything along the way. Yeah, please.
So what causes the wide spectrum here? Do you treat the electron differently?
No, the electrons basically the electrons have a critical energy that's the highest energy that they can emit from, but they can also emit the waves of any longer wavelengths. Any lower energy can also be emitted as well. And so it's just a factor of how much energy the electron has versus what magnetic field it's in gives you the highest it can get out to, but it can also emit lower energy light. And so it'll go all the way down, and then the low end is physically limited by how big your vacuum chamber is that's holding the electrons in. That's the biggest wavelength you can hold, is just physically that size. So that's why in the microwave it finally stops. Yeah?
Is the focusing similar throughout the range?
Yes, it's similar. I'm going to go into a detail in the infrared, which is that it's diffraction limited. And because it's diffraction limited, it is related to the wavelength that we're using. And so for the longer wavelengths, this is actually a perfect point source because it's smaller than the wavelengths that we're using. In the X-ray, those wavelengths are getting smaller than the electron bunch that we're starting with. But in the infrared, it actually is, the electron bunch is physically smaller than the wavelengths we use. So it becomes really a perfect point source.
Wow.
Yeah.
And the undulator spacings are various beam taps for different wavelengths.
So the undulators is the place where it's a little bit different. So the undulator, by having the different spacing set up, you can make it so that one particular wavelength will be in phase with the next bend and the next bend and the next bend, right? And so if it's in phase, then the electric fields are actually summing up directly, which means the intensity goes up like the number of electrons squared, because it's the electric field squared for the intensity. And so in that case, you can get a certain wavelength will get extremely intense, but then the wavelengths around that are not intense. They're just sort of a regular bend magnet stuff. Yeah. And so certain science that really needs to use one wavelength to do it, they'll go to these undulator beamlines, and you can tune the undulator, within some range to be able to cover certain energy X-rays, for example.
Not to be picky, but on that previous slide, 10 to the 13th seems a little, it's 10 to the 3rd for the light bulb, right?
Oh, sorry. 10 to the 13th times a light bulb is what this is.
Oh, okay.
Sorry, that's just a funky notation that somebody else used, and I stole this slide from. Yeah, no, so this is, yeah, exactly. This is how much more this is than these sources.
The numbers get bigger.
Yeah, the numbers get bigger as you go up, yeah. Yeah, these are like unreadable on the side, but.
Thanks.
Yep. Okay. So as I mentioned, it's this continuous spectrum. So let me show you what the spectrum looks like, and then I'll show you the region that I'm working in. So if you just pull up the website for the Advanced Light Source, for example, the place I work at, you can find this on the website if you go into, you dig into what's the spectrum of a bend magnet, for example. And you can see that this is plotting a couple different things. It's plotting brightness, which is the thing that I was telling you most about. It also plots raw flux, and each one of these has some funky units in terms of number of photons per second, per milliradian, per angle, all kinds of things. But basically, this is a way to be able to compare the different sources, in a nice way to be able to see what's going on there. And so for the ALS, we run at this 1.9 GeV energy electron. So that means that with the magnets that we use, that tells us what's the highest energy we can reach. And so that's why this curve is going up and up and up, and then it just plummets after we get up to this critical energy. This critical energy is right up here. This is the highest energy we can efficiently produce. There's a few photons that get kicked out a little bit higher, but they're rapidly dropping off. But on the other side, there's actually lots and lots of photons over here. So most people look at this peak and say, okay, great, this is the place to work. You know, you can use this energy range. So in this case, this is 100 eV to 1,000, 10,000 eV, right? And so that's one way to do it, is to be able to use the very peak of it. But actually, you can see that we're actually only dropping off some if you look at the brightness here. So this is going down by a factor of 10. This is now another factor of 10. We still have these huge factors to start out with compared to conventional sources. So you can still do a lot of good science as you go down further. And so, you know, to me, one of the funny things about these types of plots is that it goes down and it goes to 10 eV and 1 eV. And so 1 electron volt is here. That's just a little below the visible, right? That's just barely scratching what I call the near infrared. It's not even in the mid-infrared, which is where we can really do all the spectroscopies. And so these curves actually keep going, so I'm just going to draw some lines here. And that's really still correct. They'll just keep going along here. And so this is that whole infrared region, right, that actually, you know, keeps going on out here. You can see in terms of the total number of photons, we're only down by factors of, you know, 20 or something like that from the peak. But we're still getting a lot of photons that are out here, and we're well above where this final cutoff in the microwave finally happens. And so what I'm going to tell you about is actually this part. There's, at the advanced light source, we have 40 beamlines around the circle, and so I run 2 of them that do this. The other 38 are all using this region. And so I won't tell you basically anything about those. I'd be happy to later if anybody would like to. Or, you know, feel free to come by Berkeley anytime and get a tour of the ALS. We do that all the time as well. So let me jump to infrared and just kind of briefly step back and say, what is infrared good for, right? Why do people use infrared as a spectroscopic technique at all? And so let me jump way back. So to William Herschel. This is just a really tiny history lesson. How did we figure out that there is infrared at all? By this time, people had figured out that, you know, light has a spectrum, that there's that there's different types of colors in light and so forth. And so Herschel just did a very simple, clever experiment. He took a prism. So this is actually the prism he used from that's in the Science Museum in the UK now. And this is a sketch of what the experiment he did was. So basically, he just took a prism, he let the sun shine in, it created this rainbow color here, and then he would just turn the prism and then look at some thermometers here and say, okay, he wanted to know which of these colors was the hottest color, right? So what intensity does the sun have in the green and the blue and the yellow and so forth? And you could figure out something about the sun maybe. So that's pretty clever. And what he found was that as he was turning this, he goes down through the rainbow, and he gets down to the red, and it's going up, and he gets down past the red. He can't see anything anymore on the thermometer, and it's still going up, right? And it's still getting warmer. And so then he keeps turning it a little bit more, and he sees even more temperature that's down in there. And so, of course, we know this as infrared, and so infrared actually comes from a word that means below red, because they didn't know what else to call it, so it's just the something past the red spectrum. And so this was a clever experiment that said that there's something down below there. We can of course feel the heat from the sun or from other sources with our skin. We're actually a pretty decent infrared detector ourselves. And so you can feel the heat going down into this infrared region. But now what I want to do is tell you, so this is neat and it's a nice little history, but what's it really useful for in the scientific terms? And so there's a bunch of different things that you can do with infrared spectroscopies and a bunch of different types of sciences, but the most used part of it is the fact that if you think of balls and springs, if you think of atoms as those balls and atomic bonds as those springs, so single bonds, double bonds, whatever, different spring constants, then this is a very simple physics equation. You can solve it. Spring constant, mass that's involved. You could think of this as like one hydrogen sitting on the side of a large molecule, so that it looks almost like a wall on one side. Or you can of course work it out for two masses with two different different types of spring constants, all kinds of things. Now, that's really useful because if atoms vibrate with specific frequencies, and the specific frequencies are related to what bonding they're in and what mass they have, that means we actually are pretty sensitive to which atom is there, as well as if it's in a single bond or double bond, type of environment. And so if you can then spread this out and be able to look for these absorptions as you're going along, you can start to then match the spectra that you can measure to huge databases that have been built up over the past 50 years that can tell you what you have, what molecule is there. And so the first thing that people will use is you use an infrared spectrometer. You can put in a miscellaneous powder or just a gas or whatever, and you'll see very quickly what types of materials that is made of, right? And usually for things like CSI shows and stuff like that, they'll actually sprinkle some random powder on there and they'll say, aha, this is the killer left this for whatever reason. And oftentimes they're actually using infrared spectroscopy as the thing underneath it, but they don't tell you that. But it's so this is actually one of the most powerful pieces of it. So if you can then identify what's there, then you can find out what unknown compounds are, but you can also start to do things like look at how chemical processes are changing. You can now start to think about biology, because that is a lot of complex chemistry that's changing. You can really start to look at a lot of different pieces of it as it's going through, and just look at this infrared spectra as a way to find out what's going on inside these different spectra, what's going on inside these different molecules underneath. So let me give you a quick example as to what it is, just to kind of give you a little background. So I'm going to start with something very simple. So CO2. This is a simple molecule, and so CO2 has a few ways that it can vibrate. So I've put them down here, but let me talk you through them real quick. So this is you can call this normal mode analysis or the eigenmode analysis, or you can do all kinds of different ways of analyzing this mathematically. But basically, what you're asking are what are the fundamental motions and vibrations that this type of molecule can make. So there's some that I didn't show here, right? The whole molecule can just move to the left or something like that. That's just a translation. So that doesn't have any restoring force, so there's no vibration piece to that. So I'm going to ignore that. So there's up, down, left, right. It can flip around its long axis or this other axis. Again, that's something you can just do freely in a gas, and it will just go and do it. So again, that's a detail that doesn't have a restoring force, so it doesn't have a vibrational type mode. So let's talk about now things that have a vibrational restoring force. And so if you look at oxygens and carbon, you can think of the different possible motions that you can have, and the normal modes of them work out into these three different things. So the top one, if you look at my head is the center carbon, and the oxygen are my hands. Right, so this is now symmetrically stretching this bond, and then it comes back, right? So it's going back and forth like this. The important piece to this part is that the center of symmetry is preserved around the center of my head, right? Because if my hands are out, the symmetry is all exactly the same. If my hands are in, the symmetry is still exactly the same. It's all around the center of my head. Because of that means that there's no net electric field being made. So if you now put little plus and minuses on these different atoms, which they actually are moving electrons around. So that one's a little more electropositive, one's electronegative. So if you put little pluses and minuses on my hands, you'll see that there's no net electric field. If I'm like this or like this, it's always the same. It's always zero centered around the center. Any negatives are always balanced by the other negative on the other side. It's all symmetric there. So then let's talk about the next one. So the asymmetric stretch mode. This is where now, as is implied, the asymmetric it's no longer symmetric. And so in this case, now I'm moving back and forth like this, and you see that I'm having to move my head back and forth a little bit too, because I need to preserve the total center of mass. Otherwise, I'm translating, so I'm not worried about that. And so if I move my two hands this way, I have to move my head a little bit this way to preserve the total center of mass of this molecule. But now if I stop like this, there's clearly an electric field that's being created, because now I've got much more of my head charge over here, much more of my hand charge over here, and so there's a net electric field that's being put on between these two here. And it's an oscillating electric field, because you go like this and then you go like this, right, as you're going back and forth. And so now this electric field is flipping from this direction to this direction back and forth. And so that means that if we put an oscillating electric field of the same frequency on this molecule, we can drive this motion. And so that's the absorption that we're looking for in the infrared. Oscillating electric field is just another word for an electromagnetic photon. And the electro part of the electromagnetic photon is the part that we will oscillate this molecule with. And then there's a third one down here, which is the bending mode. So in this case, we can now go in the other plane. So in this case, I can go like this. I can bring my head down, my hands up. And then that now has a restoring force that's going to do the opposite this way. And so we can do this little dance, which is always fun. And so you do this little dance, and now again you have this asymmetry that's happening. You can have a net electric field because my head is more up and my hands are more down. So now there's a little electric field I can put this way, and vice versa when I put it this way. And so that again is an electric field that's oscillating, and we can again go directly to with an electromagnetic wave. So putting that together, this mode is something we cannot measure directly. It's not infrared active is the fancy way of saying it. This mode we can, and so it does have a dipole active moment, and so it is infrared active. And this one again has a dipole moment, and so it's something we can measure. So I show you here, this is a real measurement of CO2. Basically this is just somebody's exhale, and then we subtracted away the water, so you don't see that part as well. But that's just a simple source of CO2. And indeed, you can see this is the asymmetric stretch here, this mode here. This is the bending mode that's down here. The bending mode is lower energy than the stretching mode because it's now bending these springs instead of going directly on the restoring force of the springs. So this has a much higher spring constant than a bending of a spring would have. And so this has a lower frequency, this has a little higher frequency. And in between is where this one should sit, and in fact there's nothing. We don't see anything there at all. And so it really does work like this. We can then look at these modes, we can find out exactly where they are. There's all kinds of little subtle details, like if you look carefully there's actually 2 peaks here because there's actually different masses of carbon that are naturally available, things like this. So there's some details in there that we can get into, but basically this is a really nice way to be able to do a quick measurement of whatever is in your material by just looking at these vibrations of the bonds. And so then I won't talk you through the details, so here's this part, but I've done those little dances for you. This is a complicated molecule, so I'm not going to talk you through this, but just to show you, so if you get to now a molecule that has more than just 3 atoms, right? So this is now a toluene atom, so usually you draw it like this in a chemistry class, but you know that on each one of these is a hydrogen hanging off. And so this is now the 3D structure of this toluene molecule. And if you look, there's lots of ways for this to move that you can absorb, right? And this will end up having this very complicated spectrum here, so we can absorb at every one of these frequencies. And for kind of the major ones, I've put a little tiny movie here that's going back and forth. And so you can do a theory of it and work it all out. But practically what's really nice is that now this becomes a fingerprint of this molecule. If we see this exact pattern with these relative ratio, these peaks and so forth, we know right away that that's a toluene. If you see a little different pattern, then you can say, okay, that's not a toluene. But if it's only a little different, maybe you say, well, it actually has a benzene ring and there's something else that's related. And so you can really start to put together different types of things. But we often call this the spectral fingerprint region because this is really unique for all the different molecules that are out there. And so it's a good way to be able to find out what you're doing. Now, if you do something even more complicated, I'm going to stop drawing little movies. Something like biology, right? So this is now a living cell. And so a living cell, of course, has millions of different molecules in it, all kinds of different things, but it has some very similar things that are repeating over and over and over. So even though if I took this spectrum and I tried to add up every million different type of molecule that's in a living cell, I'd eventually get to this. It would actually be a long process. But you can start to recognize a few things that are really related. So I plot here these 2 peaks, we call them amide 1, amide 2. This is part of the backbone of a protein. So a protein has the same backbone repeating over and over, and then it has these different nucleic acid groups and things sticking out. And so these main backbone peaks show up very nicely. DNA, RNA has some basic backbone peaks. Any types of lipids have a lot of CH groups, so those show up nicely. And there's always water that's in biological systems. So these are the biggest things you can see. There's of course a lot of more subtle little features in there, and indeed like the exact position of this peak can tell you something about the structure of the proteins that you're looking at. There's a lot of other subtle details in there. But I just want to show you that even for something really, really complex, we can still start to tease out some really interesting, interesting types of information that as you're coming along through here. So with that, let me pause for a second and see if anybody has a question about how infrared spectroscopy works a little bit. Yeah.
On the previous graph, you showed where the peaks were predicted to be and where they were, and they didn't agree. And CO2 is a fairly simple molecule. Why don't they agree?
Sorry, you mean in this one?
Yes.
So actually, no. You mean this predicted and observed value here?
Yes.
Yeah. So they don't agree for it's the subtleties of the anharmonicity of the bonding because it's not a perfect spring. It actually has a little bit of an anharmonicity in it, and the details of the model will change that a little bit. So this is this is using one style of prediction. There's of course a whole bunch of other theoretical styles. So you can see you get it basically right, but not exactly right. And it comes down to these really subtle effects in terms of whether this is in a gas with other molecules nearby or other sorts of things, or if it's in sort of a pure vacuum, then it's a little different. And then the models have all of these slightly different modeling as well.
And just to help, the 546 inverse centimeters, can you convert that in your head to nanometers? Is that optical, or that's still in?
Sure, this is all in the infrared. Sure, that's a good question. So 1,000 wave numbers is 10 microns, right?
10.
So 10 microns is pretty long in the nanometer scale, right? So 10,000 nanometers in that case. 2,000 would be 5 microns. 3,000 is 3 microns. It's a 1 over effect for there because we use this funny unit, this 1 over centimeters unit. But yeah, basically energy is going up as you go along that way. So optics is all way down here somewhere, way up there. Yep. Okay, so then let me come back to the synchrotron. So now, when you have a synchrotron light source and you can do infrared spectroscopy with it, why would we bother to use this big machine that's really made for making X-rays and all this hard X-rays and soft X-rays and all these other things? Why would we actually put this on for doing infrared as well? And so the main reason is this fact that I alluded to, that this is diffraction limited, that this is actually using the electrons are physically smaller than the wavelengths that they're actually emitting in the infrared regime. And so that means that the size of the focus spot that we can put back down is as small as the wavelength of light. So I'll show you that several different ways as we go through this talk. But this is a real measurement that I did, where basically I just made a smaller and smaller holes, right? So an aperture that's getting smaller and smaller in size. And so we can start with something that's, that you can almost see up here. Thickness of a hair is typically 100 microns kind of. So a little bit below that, and then getting on down. And so as we go down further and further with a typical infrared source, this is usually a thermal source. It's like a light bulb. It's basically just something that's a hot piece of metal. To be able to measure something smaller, you can just close down your aperture. You can close down the size of your sample, and less light goes through. And you can see this is on a log scale, so you're really going down in your total signal to noise that you get at the other end. So I can lose a little bit in signal to noise and still be able to do a good measurement if you average longer, for example. But if you start losing huge amounts, factors of 1,000, then you really get to the point where you just can't do the experiments very well at all, if at all. Whereas the synchrotron, as you see, if I start putting my aperture, my sample size smaller and smaller, you can see that actually nothing changes as you're going smaller and smaller until you finally get about here. So this is about 20 microns in size. And so 20 microns is 500 wave numbers. It's the very edge of the spectrum. And so that's where we're just starting to clip the edge of this spectrum. As we go to 10 microns, we're now clipping a little bit more. That's 1,000 wave numbers of it. And then we start clipping a little bit more. So basically what we're doing is we're starting to now touch the physical size of the wavelength, so you get to the point where you can't transmit through a hole anymore of that size. And so we're just starting to now actually physically cut off the wavelengths that are possible to go through. But the key point here is that if you sit at something that's fairly small, say 10 microns or so, you're winning by a factor of 1,000. So that's really the big advantage of being able to do this with the synchrotron source. So if you can go to a factor of 1,000, then you get this fully diffraction limited spot size. The spot size we can actually measure. It's in this kind of 2 to 10 micron region is the simple numbers to keep in your head. And you can do all kinds of different things. I'll give you a couple really quick examples, and then I'll go into a couple more interesting examples, and then we'll jump into how can we do better. And so that's sort of the idea for the rest of the talk. So quick example. So here's one, a living cell. So I already showed you a spectrum of a living cell. This is what a cell looks like under a microscope. These are some cells that are under a visible light microscope. You can see they have a few different sizes because they're in different stages of growth and so forth. But if we wanted to measure, just one of these cells, we want to be able to get basically an aperture, or we want to, only have a beam that's the size of one of these cells, and that's all. And so you can, as I said, you can cut down your aperture with a traditional source. So Globar is the name of the traditional infrared source. And if we cut it down to the 6 by 6 micron region, we're just kind of barely able to get any light through here. So that was way down on that plot. So this is what that signal noise looks like when you actually do a real measurement here. And so if you look carefully, there's 2 peaks here. That's the protein peaks. There's some water here. That's the water peak. But you really can't see anything else, and even those peaks are pretty crappy. And this is a fair bit of averaging. This is many minutes of averaging. And so this is the point where you say, no, you just can't do this measurement. If you take the same sample and you now use the synchrotron source, so the synchrotron with the same apertures left in, you can see we get very nice signal to noise immediately. We didn't have to average hardly at all. And you end up getting an extremely good signal to noise. You can see all the spectral bands and features that you want to see, whereas here you just couldn't see it at all. So this kind of ability to do science that you couldn't do before is really the point of a lot of the synchrotron beamlines. Here's one more quick example. So this is studying materials under very high pressure. So this is trying to understand geology at the center of the Earth, for example. There you need to have extremely high pressure, and to do that, the way you do it is you put it in what's called a diamond anvil cell. So these are 2 diamonds that are facing each other. You press on these 2 diamonds. Right in the very center of that is there's a little hole in the gasket. Your sample loads in there, and you press as hard as you can, and until the diamonds crack, you can get extremely high pressures. That's right in that very center. Now diamonds are pretty easy to use for the infrared because they're actually fairly transparent because they're just one carbon all the way through. And so we can actually measure right through them. And so this was some work that was several years ago done on the Berkeley campus where they were trying to measure this new type of mineral. And trying to see where is it going under pressure. And so they did this measurement under pressure looking at these OH stretch region. And so if you look here, this is the best they could do. They actually averaged a huge amount to be able to try to do this. They came to the synchrotron, and you can see right away, they could start to see these peaks very nicely. Now this is a very small region of sample through diamonds, in a complex system. So it's a little tricky, and having a source that you can then focus down to a point really nicely makes a big, big difference, because you can just throw that right through this complex setup. And then I didn't, tell you the exact details of this. It actually took this poor graduate student, more than a day of, working on this stuff. They did a full 16 hours of averaging all night and stuff just to be able to pull out this crappy spectrum. At the synchrotron, we did it in 2 minutes. So when they figured this out, then they basically lost their capability of using their lab bench because they just stopped using it. It just really wasn't worth using anymore for all the struggles that they've been doing. And so now they're, all their graduate students and a whole lot of other high pressure people, they'll come to beamlines like mine and just bring their diamond anvil cells and be able to do measurements much, much faster and much, much higher quality to be able to see what's going on. So those are my 2 really quick examples, just to tell you that Berkeley's not the only place that has this. There's actually these big $100 million light bulbs all over the world. There's one in Canada, there's kind of 4 main ones in the US, a whole slew in Europe, a whole slew in Japan, and then a few others sprinkled around the world. And so what I'm plotting are only the ones that have infrared beam lines, and so these are the places where you can go to do infrared science. There's some more that are only doing X-ray science. But they're really doing all kinds of different types of science from all different types of places. Of course, if you're here, you'd probably come to Berkeley because it's the closest one. So with that, let me jump to a couple examples where I'll spend just a few more minutes kind of showing you some of the cool high-impact science that we've been able to do. And then I want to ask the question, how can we do even better? Okay, and so we'll jump into that part for the last kind of half of the talk. So the STARDUST mission is something that NASA flew now almost 10 years ago it landed, so 8 years ago it landed, back on Earth. And so this was a really cool mission because this is a mission that flew through the tail of a comet. So this is the little artist's conception of how this worked. As it was flying through this tail of the comet, it opened up this capsule, put up a catching device. So that's what this picture is here. In that catching device, they hoped to capture whatever dust stuff was making this big plume of that we could see, visibly coming off the back of the comet. And then they close it back up. And then when it got back to Earth a couple years later, they jettisoned this little part, and that's what comes down with a parachute, landed in the Utah desert. And so these were actually the first sample return missions since the lunar mission, since going to the moon. And so this was a big deal, just to have stuff coming fresh from a source way outside of the Earth. And so this was a big deal. So when these samples came in, they were all opened in this clean room. It was a big deal to be able to try to pull them out of this material. This is this stuff called aerogel, this extremely light material that could take the heat basically of stuff suddenly stopping, going from a slow speed of so many kilometers per second. I forget what that is, but to them that's very slow. And then being able to stop it, catch it, and then return it to Earth. And so samples were then cut out very painstakingly out of this aerogel and sent to different labs around the world to study them. So there's a lot of studies that people wanted to do. They wanted to be able to look at the mineralogy. They wanted to look at what kind of components of different things are in there. A lot of it required like slicing up the samples, doing electron microscopy, or doing X-ray spectroscopies where the X-rays can damage the sample because these are very high energy photons. And so one of the first places they took it was our infrared beamline. So this is me a few years ago. And so because we wanted to be able to look with a very sensitive but also very gentle tool. Infrared photons are very low energy, so they don't hurt anything that's in there. And so if there are other little things like organic molecules or other sorts of molecules in there, we wanted to be able to find them by using a gentle tool first and then progressively build up to the ones that slowly destroy the samples. So we did a whole bunch of measurements, and so this is just one slide that came out of some of it. This is the type of section that they would cut out of the aerogel. And if you look here, this is the captured track. So this is where a particle hit. The particle kind of broke apart as it did this, first kind of violent hit. And then you can see this track that goes all the way down here, and then there'll be a particle way down here is the remnants of, the particle that entered from over here. And you can see even some of it broke off and went a little bit this way. And so you can go in and now find these little particles. And so we did measurements looking for those particles. We did a whole study of those particles, saying what kind of minerals they were in, all kinds of other stuff. What this is actually showing is some of the other interesting things we found. And so one of the other interesting things we found is that in some tracks, so like here's a track, you can see this is the track coming in now from left to right. If we look at the infrared of the particle at the end, we did that, but then we started looking all over. And if we look at this, the total aerogel all around, we did a map. So this is where we take a spectrum from every point and then we plot the result of that spectrum basically. So what we're plotting here is the CH stretching. So where do we find CH2, CH3s kind of molecules? So these are anything that has an organic chemistry kind of background to it will show up there. And you can see what happened is that as this particle entered, this particular one blew off a whole bunch of something, OHs and CHs and other stuff that got blown off in this first part of the track. The particle ends up way down here, but there's quite a bit. And this is different than many other tracks. They usually look like this, and this is what this infrared map looked like. You don't really see anything there. If you look really carefully, you can see a little bit. But on the same scale, there's really nothing there. And so a few of the tracks had these huge plumes of other stuff. So these are ones that probably had ice around the particle, and that ice suddenly sublimated, and the stuff that was trapped in the ice is now what's trapped in the aerogel. So we did a bunch of studies on samples like this to be able to understand what other stuff was there even before the particle finally stopped as it was decelerating in this aerogel. But this was a really cool project to be a part of, and in fact, we're still doing measurements now, almost 10 years later, because now what they were looking for are the flip side of it. So in other words, they flipped around this particle catcher, and when they were just in the kind of the quiet parts of deep space, they just turned it around and let it fly for a while. So that was looking for things that were just coming off of the solar wind and other things that are just out in the solar system around us. And so in that case, the particles are tiny, really hard to find, and so those are way, way, way rarer than these. These are already pretty rare to be able to get them in the lab. But these other ones are now something that people have looked at very, very carefully and are trying to pluck them out. So that's actually what we're currently working on for this particular project, is trying to identify these types of materials. But this was one of our first results that came out here. The other kind of big science thing that we helped on was when there was this Deepwater Horizon explosion. This was a BP oil well in the Gulf of Mexico, and so if you saw any TV coverage or whatever in April 2010, you saw all about this. This was a giant disaster going on, and these are a few pictures showing this exploded well that's up on the surface, and then trying to figure out what to do with it. But on the bottom of this, so at the bottom of where the pipe goes down under the ground, this is now how the pipe comes up, and this pipe got just sheared off. And so this is oil just gushing out directly from the oil reserves below and into the Gulf of Mexico water. So this is a disaster. This is something you really, really don't want to have happen. And it wasn't turning off. It took them a long time to figure out how to actually finally cap this off. And so they were leaking something like 65,000 barrels of oil per day, and this went on for nearly 90 days before they were able to finally cut it off. So this is a huge amount of oil. And so one of the things that we got involved with is trying to understand what's going on inside that oil. And we sent a bunch of postdocs, a bunch of graduate students out on boats and be able to pull up actual samples. And so these are some of them. We had these special tubes. So basically, as you drop these down under the subsurface, you can, at a certain depth, you can open up each one of them and get some sample of the water that's there, bring it back up, and do some analysis. What we found was way down deep. So this is 1.2 kilometers deep. There was a huge plume of oil that was down in there. It was I forget, 15 kilometers long, a couple kilometers wide, and 1.5 kilometers down. Was this huge plume of oil that was down there, and we wanted to find out what was going on in that oil. Why is it there? But also, what else is going on there? We do a lot of looking at biology, and so we wanted to say, are there some bioactivity that's happening at this huge amount of oil that's down in there? So samples were flown every day back to Berkeley, and then we'd start looking at them under the microscope. So if you look kind of carefully here, projector is a little hard to see, but in the circled regions are a bunch of little bacteria sticking on the side of this big droplet of oil. This is in water otherwise. So if you see here, there's one there, a little hard to see. But these bacteria are actually stuck onto the side of the oil droplets, and so they seem to be liking it there, these little microbes that are there. And so we did all kinds of analysis of those microbes. We did gene sequencing. We did a whole bunch of other things. These are a few of the pretty pictures we got from them. But from the infrared part, we could actually watch these now over time. And so this is just a one-slide summary of some of the cool results that we saw. When we looked at just a very small region, so this is now a visible microscope image that has these little kind of black blobs, these blobs on the background are the oil droplets. So these little droplets are kind of scattered all around on here. And what I put on top of it is a heat map. So basically it's the infrared intensity, as we've done a map over here. And what I'm doing is I'm plotting, in this case, the protein stretching, in this case, the carbohydrate stretching, and then I'll tell you about these. And so what you can see is that here we can see there's a blob of bacteria that are sitting right over this oil. There's another blob that's sitting over this blob of oil. And we know it's bacteria because it has proteins and it has carbohydrates both in it. So we know that it's alive. It's got some kind of living system in there. And so these are the microbes that we could see under the microscope. When we then looked at what was happening by just looking at the different spectra. So the different spectra, we could just then plot different regions of what's happening. And so we've studied these bacteria over time, and what we can see is, for example, the oxidation breakdown products of oil, so the bacteria that like to eat the oil and use an oxidative path, these ones started making a lot of these oxidative products. These not so much. Whereas on the alkane side, these ones did a little, but these did a lot, right? And then down here, the nitration products got really strong on the top part, not as much here. And again, sulfates kind of has the opposite components. So what we could see is there were actually two different families of microbes here. One that like to use these oxidation and nitration breakdown pathways, another one that uses alkanes and sulfate breakdown pathways. And so they're actually eating these oil, breaking it down over time, and being able to use that as a type of food source. So this was a little bit of dumb luck, right? This is the fact that we're having an oil spill in a place where there's oil naturally seeping out of the ground. That means that nature has evolved some bacteria that like to eat oil. And so these bacteria suddenly had the all-you-can-eat buffet. They went just crazy with them, and so they multiplied. They had tons and tons of bacteria started to show up. And they were able to break down this oil. And what was really cool is we could have our little test tubes, and you could see this kind of oily water. And then you just, you leave it on your desk, and 2 weeks later, perfectly clear. You can't see any oil at all, right? And so we, of course, did the science following it as it was going down. But 2 weeks was the magic time scale for being able to eat up this oil, whichever pathway they used. That seemed to be about the mean time it took them. And so what was really heartening was that, 87 days later, they finally capped off this well. So there was this huge plume of oil down under there. We kept sampling it every day and so forth. And again, 2 weeks later, that whole plume of oil was gone. The whole thing underneath the deep subsurface was actually eaten up by these microbes and bacteria that were there. And so that was lucky. Sorry?
No, nothing. I was surprised.
Yeah. So that was lucky that they were there, but also remarkable that they could do it on this kind of scale. That this could really actually work on this this scale of this type of oil. Now, of course, there was still oil that made it to the surface. It was still all kinds of a disaster, but it could have been so much worse if we didn't happen to have these microbes that were here. And so this was something that we did kind of, after the fact or in the heat of the moment, so to speak. We started to study all this. We didn't know exactly what these bacteria were doing there. And so the follow-on studies to this have really been to now start taking samples at well sites all over the world so that we can maybe have some idea of what to do if something happens again. We can at least start to say, okay, well, certain parts of it can be taken care of, other parts of it we need to help take care of, other sorts of questions like that. So that becomes a whole different way above my pay grade kind of questions. But we can supply some of the science answers underneath it to be able to talk about it. Okay, so I've gone on and on here. I wanted to show you just a little bit about what can we do next. Okay, so I've told you about diffraction limited spectroscopy. This is where we've done all kinds of science, and I've only showed you a couple cool examples. Where can we take this beyond the diffraction limit? And so there's two new directions that we've pushed. One is to go into the third dimension to make imaging now, instead of these 2D pictures, into a full 3D picture. And then the last one, which I probably won't have time to tell you about, but I can tell you later if anybody's interested, is going down into the nanoscale. So really breaking the diffraction limit, getting down to where we get down to the nanometer level scales, but still using these micron scale wavelengths. So that's been really fun. We just got that working this year, so this was our paper about it. This microtomography we got working last year, and so it's something that's a new direction for us. So for me, this is where are we going with the synchrotron infrared technique at all? This is kind of my answer, is that we want to keep pushing towards new directions. And so I hope this will kind of spur on new types of science that I haven't even thought of at all. So firstly, let me tell you a little bit about the tomography. This was a collaboration between my group in Berkeley and Carol Hirschmugel's group in Wisconsin, and then we got samples from a bunch of other different people. Let me just take a quick step back and tell you what tomography is. Tomography is a way to measure something in the 3D, but it's a little bit tricky. Okay, so let me just show you the idea. So if you look at a 3D object and you shine light through it, what you'll end up getting is a projected image on the other side. So this gives you a shadow image, basically. And so that shadow image is something that gives you all kinds of information, but you really can't see which is in front of what, and is it a circle or a cylinder and so forth. So what you want to be able to do is to be able to actually find that 3D image. So it's just the similar way that we use two eyes to see in 3D, because we have two different perspectives. We want to get multiple perspectives to be able to build that up. And so very simply, this is an example where you have three perspectives of one thing. Shows you that there's something here, there's something here, there's something here when you look at it in these three different angles. We can do what's called back projection. So we take that thing and that tells us that something is on this line, this tells us something is on this line, this tells us something on this line. So we start to build up that there's something in fact there. And so if you do that with 100 angles instead of 3, then you get a picture that actually looks very much like the original source. So now if you want to do this with all of this type of system, you can now start to reconstruct a much more complicated thing, and then now virtually in your computer, so not actually slicing the samples, but slicing just the virtual data set, you can find out like this slice has got these two things in it, this slice has got these two things in it, and so forth. So that's how CT scanning works in medical imaging, lots of other types of places that people use computed tomography. Now, because I'm a spectroscopist, I see this as great, but it's all still grayscale, right? I really want to say, what are those molecules? And I've been telling you, infrared's great for identifying molecules. So this is what I want to do. I want to do it in full color. I want to be able to say, you know, this red thing, I know it's red because it's got, you know, some particular chemical in it. And so to do that, we had to have a couple new things to do. One is a fast instrument that could give us the big images fairly quick, so I'm saying like in a minute kind of time scale, so we can do 100 of them in a reasonable amount of time. A way to rotate samples, which is not normal for us, and a way to reconstruct the tomography. So basically we can use the same algorithms that medical tomography uses, but we need to tell those algorithms that there's actually multiple spectra involved, so it's a little bit tricky. So we built a system that could do this, and so this is just a little picture of a mini goniometer sitting in the focus of an infrared microscope here, and that little bright spot is where the sample sits, and so we can rotate some sample around and be able to now take measurements of this at every angle as we're going around this. This is one of the advantages of working at a synchrotron, is that I just walked over to my hard X-ray imaging beamlines, and they had these kinds of pieces working. I walked to the tomography beamline, and they told me about the software. And so I was able to get all of this working in a kind of quick and cheap way to see if this would work at all. So my very first sample was just a loop. And so this is a protein crystallography microloop. It looks like this on the end, the business end of it. And so when I did this full 3D spectra, so now I'm just going to plot one absorption band as a function of position. And if you look here, it actually looks 3D, right? And so to me, this was totally remarkable. This was actually even before we had it motorized. This was just me turning it by hand to see if this would work. And so the fact that we got this to work at all, and it has the right shape, this was thicker, this is thinner, all that kind of stuff looked fantastic. The other thing is that we can now reconstruct this picture for every single wavelength, and then I can pull out the intensity of one point within this structure and plot it back on the scale. So this is a very complicated way to do a spectrum, where you actually do a full 3D reconstruction and then plot the intensity back here. But what was really important to me was to see that the spectra actually is really there. So these two peaks are the big peaks in polyamide, which is what this stuff is made out of. This is the spectra that we expected. And so you can see the spectra actually looks very faithfully reproduced. The intensities are right. And in fact, the quantitative intensity is right too, if you look at the 3D space that we're looking at here. And so then this is really doing spectroscopy now in the 3D. So let me just show you a couple quick examples of this, and then I'll wrap up. So for our first sample, we looked at something that we had easy access to samples of, and so we like biology, and we all had some hair, so we just plucked out some hair, found the thinnest hair so we could do it quickly. And so this is a piece of hair that we've mounted on here. And we know from colleagues that about 10 years ago that did some nice studies, they took a hair, they would embed it in paraffin or some kind of wax, and then you slice it, and then you look at those 3D, you look at the slices, in 2D to be able to see what the structure of the hair is. So this is a big process to be able to do that, but you can do things like plot the protein, and you can see that there's more protein around this outside, less in the very middle, this medulla part. We wanted to say, could we do the same thing? So this is now, we took this hair, rotated it around, reconstruct the protein peaks, and you can see here that it looks like a cylinder of protein, so that's not super exciting. But if we look at it from the other direction, that's where it becomes interesting. So So now we can just rotate this in the computer, so we didn't do this on the real sample, and say, do we see this? And in fact, yeah, look, there's less protein in the middle than there is around the outside. Now, in no case did we have that view, right? We were always measuring it from the side. So the fact that we could pull out the real 3D structure of a buried object here means that this is really starting to work. And we can even say, okay, what is in there? Well, we just looked at that paper and said it must be phospholipids. So we plot the phospholipid peaks. And indeed, you can see there's a little bit of phospholipid around the outside, but basically the center part is all filled with this phospholipids. So we can really see this, what's going on the inside of that as well. So let me just show you a couple pretty pictures, and then we'll wrap up. So this is a little more interesting sample. These are some wood fibers. This is something that people want to understand for biofuels, for example. And so this is a little twig, and we're able to measure it and plot different regions. So this is the CH groups. This is kind of filling the spaces around this fiber. If I plot here now just the lignin is the kind of the main part of the fibers that you care about in the wood for biofuels. You can see that this particular little piece has 3 little cells of lignin going up here. That's the channels the water goes through. And in fact, the CH group kind of fills the space in between them if we put them together. You can see there, or if we put it on end, that the CH groups is kind of all the organic stuff in between, and then the lignin channels are showing up very nicely there. Now, the scientific question is cellulose and lignin are the 2 main components that are there. Cellulose is the hard to break down part, lignin is the energy source you want to get to. And so they wanted to understand exactly the 3D nature of this. And so if you look at now and just plot the cellulose and the lignin together, then you see that it looks like it's all the same. There are these 3 channels and they're all showing up. But now if I take the data and put it on end and then virtually cut down through the data, you can see what's really happening here. And so then if you look at this, you can see that here the greenish color is always on the inside, the reddish color is always on the outside. So the cellulose is the outer part of the channels, which is why it's hard to break it down, and that's the part that's hard to get to the biofuel part on the middle, on the inside. So, I know I'm running out of time, so with that, let me just jump to one more just kind of pretty picture of a multi-component system. This is a nanocellulose fiber composite that has 3 different materials that I'm plotting the different peaks of here in different colors. And you can look at the way this structure comes together. It becomes a natural kind of glue. And so we can look at it in all kinds of different ways, putting the system together in 3D, putting it back together now, one component at a time, to really identify where each of the pieces are, and really do some kind of new types of science there. So with that, I'll just let that play, and I'll stop here. So this is where we're pushing forward into going way beyond the diffraction limit, but by doing things that are really well beyond what's been possible so far. So with that, thank you very much.